Chukwuma Chinaza. I'm in New World Academy SS2. I'm from Science Department, Biology, precisely Group C. I am here to enlighten us more about a biology concept, dissection. The study of mammals is a very important part of biology because human beings are mammals, and in studying a rat or any other mammals, we are to a large extent studying ourselves. Here are some of the apparatus needed in dissection. We need the dissecting board, the dissecting pin, the forceps, and the scalpel. The procedures for dissection are: we place the animal on its back to the dissecting board using your dissecting pin to a full extent. Forelimbs and the hind legs on the board are hold or held with the dissecting pin. You might be wondering why we use the animal we are going to use for this dissection. We choose to use the chicken because it is the one that is commonly found around our environment, as it is the one that we are going to be easily seen closely. After placing it on the dissecting board and holding it with the dissecting pin, we slit the skin through the center of the chicken and then to the level of the anal, so that we are going to be able to study the internal organs properly. In the chicken, here are some of the internal organs. We have the small intestine and the large intestine. The large intestine is also known as the colon, while the small intestine is known as the anus. We also have the backbone, which is also known as the vertebral column. We have the liver, we have the stomach, we have the trachea, and etc. So the chicken is considered as a mammal most time because it has some of similar characteristics of that of mammal. It has the internal organs related, which is similar to that of the mammal, such as the small intestine, the large intestine, the vertebral colon, etc. Now, why it is not considered as mammal also is because they don't have mammary gland and they are not able to give birth to their young ones alive. Rather, they lay eggs, and this is why they are seen as oviparous animals. After the practical, to preserve the body of the internal organs of the animals which we are going to remove from the dissection process, we use the formalin, which is a chemical used in preserving organisms, dead organisms or body, in order to prevent smell or decay. Here we have the practical we just concluded with the formalin in it to preserve it. With this, it can stay for years without getting a sticky odor or decay. Now, anatomy is seen as the study of the internal organs. That is why in dissection, the main purpose of that dissection practical we just concluded is just to study the internal organs, which is seen as anatomy. Thank you.